Welcome to The Calm Scientist. If you're here, it probably means you're ready to slow down, relax, and drift gently into the night with a head full of curiosity. Tonight, we're going to unravel one of the most profound questions humanity has ever faced. A question that sits at the intersection of our deepest scientific capabilities and our most fundamental ethical concerns. We're exploring the remarkable, unsettling, and utterly fascinating world of DNA manipulation and cloning, where the building blocks of life themselves have become our playground, our laboratory, and perhaps our greatest moral challenge. I do hope this journey through genetic engineering isn't terribly inconvenient for your plans to drift off to sleep. Though considering we'll be discussing the power to resurrect extinct species, create genetic duplicates of beloved pets, and potentially design the next generation of humans, well, you might find yourself a bit too intrigued to doze off immediately. It's the kind of bedtime story only a collaboration between Mary Shelley and Charles Darwin could write, a tale where science fiction has quietly, almost imperceptibly, transformed into science fact. Before we begin, if you're enjoying this gentle stroll through the frontiers of genetic possibility, I'd be ever so grateful if you gave the video a like and subscribed. It really helps this cozy little channel continue its mission of making complex science feel as comfortable as your favourite blanket. And if you're feeling friendly, let me know in the comments where in the world you're listening from and what time it is where you are. I love imagining our little global campfire of curious minds stretching from moonlit Manhattan to sleepy Singapore, all connected by our shared wonder at what it means to hold the code of life in our hands. This video is here to relax you, to fascinate you just enough to make your thoughts soft and meandering. You won't need to remember anything specific. Just float with me as we explore the extraordinary moment we find ourselves in, where the boundaries between what nature creates and what humans can engineer have become beautifully, terrifyingly blurred. And just before we begin, this video is for relaxation and education. It is not medical advice or a substitute for professional care. Always look after yourself and sleep well. Let's start with the basics, or what we might think are the basics. DNA, deoxyribonucleic acid, sounds like something straightforward, doesn't it? 4 simple letters, A, T, G, and C, arranged in various combinations to spell out the instructions for every living thing on Earth. It's like nature's alphabet, we tell ourselves, a biological programming language that writes the software of life. But hold that thought gently, because as it turns out, even something as fundamental as our understanding of DNA has transformed dramatically in just the past few years, particularly in 2025, where we stand at an inflection point that would have seemed impossible just a generation ago. Think about this for a moment. In 1953, when Watson and Crick first described the double helix structure of DNA, they could barely imagine reading the genetic code, let alone rewriting it. The idea of editing genes was pure fantasy, the stuff of pulp science fiction novels. Fast forward to today, and we have high school students using CRISPR technology in classroom experiments. We have companies that will clone your cat for $50,000. We have scientists who claim to have brought back extinct species, creating living dire wolves from ancient DNA. The journey from discovery to manipulation took less than a human lifetime, and the pace of change is only accelerating. Here's where things get interesting, and by interesting I mean both wonderful and deeply unsettling. The technology we're discussing tonight isn't some far-off possibility. It's happening right now as you lie there listening to my voice. Somewhere in a laboratory, probably several laboratories, scientists are using a molecular tool called CRISPR Cas9 to slice into DNA with the precision of a surgeon's scalpel, except this scalpel operates at the molecular level, making cuts measured in billionths of a meter. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, and yes, that's quite a mouthful for bedtime, was originally discovered as part of the bacterial immune system. It's a bit like finding out that bacteria had invented genetic engineering billions of years before humans even existed. These microscopic organisms use CRISPR as a way to remember viral invaders, storing fragments of viral DNA like wanted posters in a cellular police station. When the virus returns, the bacterium can recognize it and send out molecular scissors to cut it apart. But here's the truly remarkable part. Scientists have taken this ancient bacterial defense system and turned it into the most powerful genetic editing tool ever created. It's more accurate than anything we've had before, more versatile, more accessible. Where previous genetic engineering techniques were like trying to edit a book with a sledgehammer, CRISPR is like having a word processor with find and replace functionality. You can target specific genes, specific sequences, even specific letters in the genetic code, and change them to whatever you want. The implications of this are staggering, and they're not theoretical anymore. As of early 2025, we have several CRISPR therapies already approved and in use. There's Casgevy, the first CRISPR medicine approved for treating sickle cell disease and beta thalassemia. Imagine that, a genetic disease that has plagued humanity for millennia, causing tremendous suffering and early death, can now be treated by editing the patient's own cells. The therapy works by reactivating the production of fetal hemoglobin, essentially convincing adult cells to produce the healthier version of hemoglobin that we all made as fetuses. In hospitals right now, children who would have faced a lifetime of pain and medical complications are being treated with personalized CRISPR therapies. Just this year, at Children's Hospital of Philadelphia, a baby named KJ became the first person ever to receive a completely customized gene editing treatment. He was born with a rare metabolic disorder that would have been fatal, but scientists designed a bespoke CRISPR therapy just for his specific genetic mutation. They created it in 6 months, from diagnosis to treatment. 6 months to rewrite the code of life for one specific child. The treatment worked, and KJ is now thriving, growing, living a life that wouldn't have been possible even 2 years ago. Meanwhile, other forms of genetic manipulation are advancing at an equally breathtaking pace. There's base editing, a more refined version of CRISPR that can change single letters of DNA without cutting the double helix. Think of it as the difference between surgery and medication. Both can heal, but one is far less invasive. There's prime editing, which can insert, delete, or replace longer sequences of DNA with unprecedented accuracy. Scientists at Yale have developed new CRISPR tools that can edit multiple genes simultaneously, opening up possibilities for treating complex diseases that involve many genetic factors. But medicine is just the beginning of this story. Let's delve deeper into something that might feel more personal, more emotionally complex: the cloning of pets. Right now, as you listen to this, there's a company in Texas called Viagen Pets that has a 5 to 7 month waiting list for people who want to clone their dogs or cats. The price tag is $50,000 for a genetic copy of your beloved companion. Not a similar looking animal, but an exact genetic duplicate, created from cells taken from your pet's skin. The process itself is both remarkably sophisticated and surprisingly straightforward. Scientists take a small tissue sample, usually from a skin biopsy, and culture the cells in a laboratory. They then remove the nucleus from a donor egg cell and replace it with the nucleus from your pet's cell. Through a process called somatic cell nuclear transfer, the same technique used to create Dolly the sheep back in 1996, they create an embryo that is genetically identical to your pet. This embryo is implanted into a surrogate mother, and approximately 2 months later a clone is born. But wait, there's more to this story, and it's here that the moral complexity begins to deepen. These cloned pets, while genetically identical to their predecessors, are not the same animal. They have different personalities, different quirks, sometimes even different markings despite having the same DNA. It's a profound reminder that we are more than our genes, that consciousness, personality, the ineffable quality we call the self, emerges from the interplay between genetics and experience, nature and nurture, in ways we still don't fully understand. The pet cloning industry has grown dramatically, especially since the coronavirus pandemic, when isolated people formed deep bonds with their animal companions. Viagen Pets reports that they've cloned thousands of animals, not just pets, but also livestock and horses. They've even worked with zoos to clone endangered species. 2 Przewalski's horses, an endangered species from Mongolia, were successfully cloned and are now living at the San Diego Zoo. Their names are Kurt and Holly, and they represent hope for a species on the brink of extinction. Speaking of extinction, let's explore one of the most audacious applications of cloning technology, de-extinction, the attempt to bring back species that have vanished from the Earth. In April 2025, Colossal Biosciences, a company founded with the explicit goal of resurrecting extinct species, announced that they had successfully created what they call the world's first de-extinct animal: 3 dire wolf pups, born through a combination of ancient DNA extraction, gene editing and cloning. Now let's be clear about what this means and what it doesn't mean. These aren't true dire wolves which went extinct about 12,500 years ago. Instead, scientists took DNA fragments recovered from preserved dire wolf specimens, identified the key genes that made dire wolves different from modern grey wolves, and then used CRISPR to edit those genes into grey wolf embryos. The result is essentially a hybrid, an animal that looks like a dire wolf, has many of the genetic markers of a dire wolf, but is really a heavily modified grey wolf. The same company has announced plans to bring back the woolly mammoth by 2029, though again, what they're really creating is an elephant with mammoth-like characteristics: thick fur, cold resistance, the distinctive curved tusks. They're also working on the Tasmanian tiger and the dodo. Other organisations are attempting to revive the quagga, a subspecies of zebra that went extinct in the 1880s, and various species of giant tortoises that have disappeared from isolated islands. The science behind extinction is fascinating and complex. When an animal dies, its DNA begins to break down immediately. Even in ideal preservation conditions like permafrost or amber, DNA degrades over time, breaking into smaller and smaller fragments. Scientists have to piece together these fragments like a massive jigsaw puzzle where most of the pieces are missing and the ones you have are damaged. They fill in the gaps with DNA from closely related living species, creating a genetic patchwork that hopefully captures the essence of the extinct animal. But here's where the philosophical questions become unavoidable. If you create an animal that has 90% mammoth DNA and 10% elephant DNA, is it really a mammoth? If you clone a dire wolf but raise it in a modern zoo, eating commercial dog food and receiving veterinary care, is it truly a dire wolf or something entirely new? These aren't just semantic questions. They go to the heart of what we mean by species, by extinction, by nature itself. The technology raises profound ethical considerations that we're only beginning to grapple with. Conservation biologists are divided on whether extinction efforts are helpful or harmful. On one hand, the technology developed for extinction could be invaluable for saving endangered species. The techniques used to edit mammoth genes into elephants could be used to give living elephants genes for disease resistance or climate adaptation. The cloning methods perfected on extinct species could help rescue species with dangerously low genetic diversity. On the other hand, some conservationists argue that extinction is a dangerous distraction from the urgent work of protecting the species we still have. Why spend millions of dollars trying to bring back the mammoth when we can't even save the elephants we have now? There's also the question of ecosystem integration. The world that mammoths inhabited no longer exists. The vast mammoth steppe that stretched across northern Eurasia and North America is gone, replaced by forests and human settlements. Where would these resurrected creatures live? What would they eat? How would they interact with modern ecosystems that have evolved in their absence? Let's take a closer look at the technical advances that have made all of this possible. In March 2025, Yale scientists announced a breakthrough that represents a fundamental leap forward in genetic engineering capability. They've developed new CRISPR tools based on something called Cas 12a, rather than the more common Cas 9 protein. These new tools can edit multiple genes simultaneously, and in different directions, some genes can be turned on while others are turned off, all in a single treatment. This might sound like a minor technical improvement, but it's actually revolutionary. Most diseases, most traits, most of everything that makes an organism what it is, involves multiple genes working together. Height isn't controlled by one gene but by thousands. Intelligence, if we can even define it properly, involves complex interactions between countless genetic and environmental factors. The ability to edit multiple genes at once opens up possibilities that were previously unimaginable. Scientists have also made remarkable progress in delivering CRISPR to specific parts of the body. One of the biggest challenges in gene therapy has always been getting the treatment to the right cells. You can have the most perfect gene editing tool in the world, but if you can't get it to the cells that need fixing, it's useless. Recent advances in lipid nanoparticles, the same technology used in mRNA COVID vaccines, have made it possible to deliver CRISPR components directly to organs like the liver, the brain, even the heart. Intellia Therapeutics has developed a CRISPR therapy that can be delivered through a simple IV infusion. The treatment, called NTLA-2001, is designed to treat a disease called transthyretin amyloidosis, where misfolded proteins accumulate in the body, causing organ failure. The therapy works by turning off the gene that produces the problematic protein, and early results show that a single treatment can reduce protein levels by up to 97%. Imagine that, one IV infusion that permanently fixes a genetic mistake. But perhaps nowhere is the combination of promise and peril more apparent than in the discussion of human germline editing, making genetic changes that will be passed on to future generations. This is the realm of what people call designer babies, though that term dramatically oversimplifies the scientific and ethical complexity involved. In 2018, a Chinese scientist named He Jiankui shocked the world by announcing that he had created the first gene-edited human babies, twin girls whose DNA he had modified to make them resistant to HIV. The international scientific community condemned his work as premature and unethical. He went to prison for 3 years, but the twins, known as Lulu and Nana, were born and by all accounts are healthy, though they're being monitored for any potential long-term effects. The controversy around He Jiankui's experiment wasn't just about the ethics of editing human embryos, it was also about the specific edit he made. He modified a gene called CCR5, which HIV uses to enter immune cells. But CCR5 isn't just involved in HIV infection, it plays roles in the immune response to other diseases, in brain function, possibly in lifespan. By editing this gene, he may have protected the girls from HIV, but he may also have made them more vulnerable to other diseases or created entirely unexpected effects that won't become apparent for years or decades. This uncertainty is at the heart of the germline editing debate. When you edit an embryo's DNA, you're not just changing that individual, you're potentially changing all of their descendants. Every mistake, every unintended consequence, gets passed down through generations. It's evolution at the speed of human decision, making rather than the slow, careful process of natural selection. Yet the technology continues to advance. Companies like Orchid Biosciences now offer polygenic screening of embryos for couples undergoing IVF. They can sequence more than 99% of an embryo's genome and provide risk scores for hundreds of conditions, from diabetes to schizophrenia to heart disease. Parents can then choose which embryo to implant based on these genetic predictions. It's not quite designing a baby from scratch, but it's a significant step toward giving parents unprecedented control over their children's genetic makeup. The cost of this technology puts it out of reach for most people. Orchid charges $2,500 per embryo just for the genetic testing, on top of the already substantial costs of IVF. This raises concerns about genetic inequality, a future where the wealthy can afford to give their children genetic advantages while everyone else is left behind. Some bioethicists worry about a new form of eugenics, not imposed by governments but chosen by parents, market-driven rather than state-mandated. There's something deeply unsettling about the commodification of human genetics, about turning the creation of life into a consumer choice. Yet there's also something deeply human about wanting to give our children every possible advantage, to protect them from suffering, to ensure their health and happiness. Parents already make countless choices that affect their children's lives, from prenatal vitamins to school districts to extracurricular activities. Is genetic selection really so different? The answer, of course, depends on who you ask. Disability rights advocates point out that many of the conditions we might edit out of the human genome, things like deafness or dwarfism or autism, aren't necessarily sources of suffering. Many people with these conditions live full, happy, meaningful lives. They argue that genetic selection based on these traits is a form of discrimination, a judgment that certain kinds of human variation are unacceptable. Religious groups raise different concerns, arguing that genetic engineering represents an unacceptable intrusion into God's domain, that we're playing with forces we don't fully understand and can't control. They point to the long history of scientific hubris, of technologies that seemed beneficial at first but led to unexpected consequences. DDT was going to eliminate malaria. Thalidomide was going to treat morning sickness. What unexpected consequences might genetic engineering bring? Meanwhile, transhumanists and other technology optimists argue that we have not just the ability but the obligation to improve the human condition through genetic engineering. They see aging, disease, even death itself as engineering problems to be solved rather than inevitable facts of life. Why should we accept the genetic lottery when we have the power to stack the deck in our favour? Why should children suffer from genetic diseases when we can prevent them? Let's examine the current state of gene therapy clinical trials to understand where this technology is actually being applied right now. As of August 2025, there are hundreds of gene therapy trials underway around the world, targeting everything from rare genetic diseases to common conditions like heart disease and diabetes. The pace of progress is extraordinary. New trials are being launched every month, and results from earlier trials are consistently exceeding expectations. One particularly promising area is CAR, T-cell therapy, where a patient's own immune cells are genetically modified to fight cancer. The process involves extracting T-cells from the patient, using CRISPR or other gene editing tools to modify them so they can recognize and attack cancer cells, growing billions of these modified cells in the laboratory and then infusing them back into the patient. It's like giving the immune system a software upgrade, teaching it to see cancer cells that it previously ignored. The results have been remarkable. Some patients with previously untreatable leukemias and lymphomas have achieved complete remission. Children who were given weeks to live are now cancer-free years later. The therapy doesn't work for everyone and it can have serious side effects, but for those it helps, it's nothing short of miraculous. Scientists are now working to expand CAR, T therapy, to solid tumours, which are much more challenging to treat than blood cancers. They're also developing off-the shelf CAR T cells that don't need to be customised for each patient, which would dramatically reduce costs and increase accessibility. Imagine walking into a hospital and receiving genetically modified immune cells as easily as getting a blood transfusion. Another frontier is gene therapy for neurological conditions. The brain has always been a challenging target for gene therapy because of the blood-brain barrier, a protective system that prevents most substances from entering the brain tissue. But recent advances in viral vectors, the modified viruses used to deliver gene therapy, have made it possible to get genetic material into brain cells. Clinical trials are underway for Parkinson's disease, using gene therapy to restore the production of dopamine in the brain. Others are targeting Alzheimer's disease, attempting to clear the protein tangles that characterize the condition. There are even trials for rare pediatric brain diseases, conditions that previously had no treatment options at all. The agricultural applications of genetic engineering are equally revolutionary and controversial. Scientists have developed crops that can withstand drought, resist pests, produce higher yields, even manufacture their own fertilizer. In a world facing climate change and a growing population, these innovations could be literally life-saving. But they also raise concerns about corporate control of the food supply, about the loss of agricultural biodiversity, about unexpected ecological consequences. The same CRISPR technology used to edit human genes is being applied to livestock. Scientists have created pigs that are resistant to viral diseases that cost the pork industry billions of dollars annually. They've developed cattle that don't grow horns, eliminating the need for the painful process of dehorning. They've even created chickens that only produce female offspring, addressing both animal welfare concerns and economic inefficiencies in the egg industry. But perhaps the most controversial agricultural application is gene drives, genetic modifications designed to spread through entire populations. Imagine a mosquito genetically engineered to be incapable of carrying malaria. Release a few thousand of these mosquitoes, and within a few generations, the modification spreads through the entire population. Malaria could be eliminated without vaccines, without bed nets, without any ongoing human intervention. The same technology could be used to eliminate invasive species, to control agricultural pests, to reshape entire ecosystems. But once released, gene drives would be nearly impossible to recall. They could spread across national borders, affecting countries that never agreed to their use. They could have unexpected effects on food webs and ecosystem dynamics. They represent a level of environmental manipulation that makes even supporters of genetic engineering pause. Let's delve deeper into the business side of genetic engineering, because understanding the economic forces at play is crucial to understanding how this technology will develop. The global gene therapy market is expected to reach over $30 billion by 2030. Venture capitalists are pouring money into biotech startups. Pharmaceutical companies are acquiring gene therapy companies for billions of dollars. Just this year, Eli Lilly announced it would acquire Verve Therapeutics for up to $1.3 billion. Verve is developing a gene-editing treatment for familial hypercholesterolemia, a genetic condition that causes dangerously high cholesterol levels. Their approach uses base editing to permanently turn off a gene called PCSK9, essentially giving patients a one-time treatment that provides lifelong protection against heart disease. The economics of gene therapy are unlike anything we've seen in medicine before. Traditional drugs need to be taken continuously, generating ongoing revenue for pharmaceutical companies. But gene therapies are potentially one-time cures. How do you price a cure? How do you ensure that companies have incentives to develop these treatments while also making them accessible to patients who need them? Some gene therapies are among the most expensive treatments ever created. Hemgenix, a gene therapy for haemophilia B, cost $3.5 million for a single dose. Skisona, for a rare neurological condition, cost $3 million. These prices reflect not just the cost of development and manufacturing, but also the value of a potential cure for a lifelong condition. From one perspective, $3 million for a cure is actually cost-effective compared to a lifetime of treatments. From another perspective, it's a price that puts these therapies out of reach for most of the world's population. The pet cloning industry operates on a different economic model but raises similar questions about accessibility and equity. At $50,000 for a cloned dog or cat, it's a luxury service accessible only to the wealthy. Yet the technology continues to advance and costs continue to fall. The price of pet cloning has dropped by 60% for dogs and 80% for cats since the service first became available. Will there come a day when cloning a pet costs no more than a purebred puppy from a breeder? Some companies are positioning themselves for that future. Gemini Genetics in the UK offers genetic preservation for just £500 plus VAT, storing your pet's DNA for future cloning when prices fall or when you can afford the procedure. They charge just £12 per month for storage, making it accessible to middle-class pet owners who might want to keep their options open. The conservation applications of cloning and genetic engineering operate on yet another economic model, one driven more by philanthropy and government funding than by market forces. Revive and Restore, a non-profit organisation, has been working for over a decade to develop genetic rescue techniques for endangered species. They've successfully cloned black-footed ferrets, created genetic diversity in populations of Hawaiian crows, and are working on projects involving everything from coral reefs to American chestnuts. The economics of conservation genetics are complex. Is it worth spending millions of dollars to clone a black-footed ferret when that money could protect thousands of acres of habitat? Is de-extinction a wise use of limited conservation resources, or a distraction from more pressing needs? Different stakeholders answer these questions differently, and the debates are far from resolved. There's something quietly profound about the fact that we're having these debates at all, that we've reached a point where the question isn't whether we can manipulate the code of life, but whether we should, and if so, how, and to what ends. We stand at a threshold that our ancestors could barely imagine, wielding powers that mythology attributed to gods. Let's explore the latest developments in our understanding of how genetic engineering actually works at the molecular level, because the details matter more than you might think. When CRISPR makes a cut in DNA, it triggers the cell's natural repair mechanisms. There are two main pathways the cell can use to repair the break. One is called non-homologous end joining, which basically glues the broken ends back together, often introducing errors that disable the gene. The other is called homology, directed repair, which uses a template to fix the break, allowing scientists to insert new genetic information. The challenge is that cells strongly prefer non-homologous end joining, especially in non-dividing cells like neurons or heart muscle cells. Scientists have been working for years to figure out how to tip the balance toward homology, directed repair, to make precise edits rather than just breaking genes. Recent breakthroughs have identified small molecules that can shift cells toward the desired repair pathway, dramatically improving the efficiency of precise gene editing. This might seem like a technical detail, but it's actually crucial for the future of gene therapy. Many genetic diseases aren't caused by genes that need to be turned off, but by genes that need to be fixed or replaced. A single letter change in the 3 billion letters of the human genome can cause sickle cell disease, cystic fibrosis, or hundreds of other conditions. Being able to make that precise correction, rather than just disrupting the gene, is the difference between a treatment and a cure. Base editing and prime editing, newer variations of CRISPR technology, bypass the need for double, strand breaks entirely. Base editors use a modified CA's protein that doesn't cut DNA, but instead recruits enzymes that can change one letter to another, like changing an A to a G or a C to a T. It's like having a pencil eraser and a pen instead of scissors and glue. Prime editors go even further, able to insert, delete, or replace sequences up to dozens of letters long without requiring a double strand break. These technologies are already being tested in clinical trials. Prime Medicine, aptly named, is developing prime editing therapies for conditions including chronic granulomatous disease, a rare immune disorder. Their first clinical trial began in 2025, making it the first test of prime editing in humans. Early results suggest the technology is safe and effective, though it's still too early to know for sure. The delivery mechanisms for gene therapy have also seen remarkable innovation. Early gene therapies relied almost exclusively on viruses to deliver genetic material to cells. Viruses are nature's own genetic engineers, having evolved over billions of years to insert their DNA into host cells. Scientists modify these viruses to remove their disease, causing abilities while retaining their delivery capabilities. But viruses have limitations. They can trigger immune responses. They can only carry a limited amount of genetic material. They sometimes insert their cargo in the wrong place in the genome, potentially causing new problems. So scientists have been developing non-viral delivery methods. Lipid nanoparticles, the technology that made mRNA COVID vaccines possible, are now being used to deliver CRISPR components. These tiny bubbles of fat can protect genetic material from degradation, help it enter cells, and can be targeted to specific organs. They're particularly good at delivering material to the liver, which is why many of the early CRISPR therapies target liver diseases. Other delivery methods are even more exotic. Scientists have developed synthetic viruses, artificial constructs that mimic the best features of natural viruses without their drawbacks. They've created exosomes, tiny vesicles that cells naturally use to communicate, loaded with genetic cargo. They've even developed ways to deliver CRISPR components directly through the skin using special formulations that help large molecules penetrate biological barriers. The precision of these delivery methods continues to improve. Scientists can now target gene therapy to specific cell types, even specific cells within a tissue. They can control when genes turn on or off, responding to drugs or natural biological signals. They can even make edits that are reversible, turning genetic modifications on or off as needed. This level of control opens up possibilities that go far beyond treating disease. Scientists are exploring genetic modifications that could enhance human capabilities, genes that provide resistance to radiation, important for space travel, genes that allow for better oxygen utilization, useful at high altitudes or underwater, genes that slow aging, potentially extending human lifespan by decades. These enhancement applications remain highly controversial and are not currently being pursued in clinical trials. But the technology to make them possible exists today. The line between treatment and enhancement is blurrier than we might like to admit. Is it treatment or enhancement to give someone genes for stronger bones? What about genes for better memory? Where exactly do we draw the line, and who gets to draw it? The international regulatory landscape for genetic engineering is complex and evolving. Different countries have taken very different approaches to regulating these technologies. The United States regulates gene therapies as drugs, requiring extensive clinical trials to demonstrate safety and efficacy. The European Union has similar requirements, but has been more cautious about approving gene therapies, particularly those involving germline editing. China, despite the He Jiankui scandal, continues to be a major player in gene therapy research. The country has more gene therapy clinical trials than any other nation and has been more willing to approve experimental treatments. This has led to concerns about a regulatory race to the bottom, with countries competing to attract biotech investment by loosening safety requirements. Some countries have banned germline editing entirely. Others allow research on embryos but prohibit implantation. Still others have no specific regulations at all. This patchwork of regulations creates challenges for international collaboration and raises the possibility of regulatory arbitrage, where companies conduct research in countries with the most permissive rules. International bodies have attempted to create consensus guidelines for genetic engineering. The World Health Organization has developed recommendations for human genome editing. Scientific organizations have created voluntary moratoriums on certain types of research, but these guidelines lack enforcement mechanisms and rely on the good faith of scientists and companies to follow them. The challenge is that genetic engineering is advancing faster than our regulatory systems can adapt. By the time regulators understand a new technology well enough to create appropriate rules, scientists have often moved on to the next innovation. It's like trying to regulate the internet in 1995. The landscape changes too quickly for traditional regulatory approaches to keep pace. This regulatory uncertainty creates risks but also opportunities. Companies that can navigate the regulatory landscape successfully have significant advantages. Countries that create clear, science-based regulations can attract investment and talent. But the stakes are high. A single serious adverse event, a gene therapy that causes cancer, or a cloned animal that suffers from unexpected health problems, could set the field back by years. Let's return to the more immediate and personal applications of this technology, the ones that might affect your own life in the coming years. Genetic testing has become remarkably accessible and affordable. Companies like 23andMe and Ancestry DNA will analyze your genome for less than $200. While these tests can't diagnose diseases, they can identify genetic risk factors and carrier status for various conditions. This information is powerful but also complicated. Learning that you have a genetic variant that increases your risk of Alzheimer's disease by 50% doesn't tell you whether you'll actually develop the condition. It might motivate you to make lifestyle changes that reduce your risk, or it might cause anxiety without providing any actionable information. The psychological impact of genetic information is still not fully understood. Some people are taking genetic testing even further, pursuing whole genome sequencing that reads all 3 billion letters of their DNA. This costs a few $1,000s today, down from $3 billion for the first human genome sequenced in 2003. The price continues to fall, and some predict that whole genome sequencing will cost less than $100 within the next few years. With this genetic information comes the possibility of personalized medicine, treatments tailored to your specific genetic makeup. Cancer treatments are already being selected based on the genetic profile of tumors. Psychiatric medications are being chosen based on genetic variants that affect drug metabolism. In the future, your entire medical care could be customized to your unique genetic blueprint. But this also raises privacy concerns. Your genome is the most personal information possible, containing details about not just your health, but your ancestry, your family relationships, even aspects of your personality and behavior. Who has access to this information? How is it protected? Could it be used by insurance companies to deny coverage, or by employers to discriminate in hiring? Some countries have passed genetic non-discrimination laws, but enforcement is challenging and loopholes exist. The long-term implications of widespread genetic testing are still unfolding. We're creating vast databases of genetic information, resources that could accelerate medical research, but also create new forms of surveillance and control. The direct-to-consumer genetic engineering market is also emerging, though it remains highly controversial and largely unregulated. Biohackers are experimenting with DIY gene therapy, ordering CRISPR kits online and attempting to modify their own genes. Some have injected themselves with genes intended to increase muscle growth or provide HIV resistance. These experiments are dangerous and unlikely to work, but they represent a democratization of genetic engineering that would have been impossible just a few years ago. There's something both thrilling and terrifying about the idea that genetic engineering could become as accessible as home computing. Will we see a future where people modify their own genes like they modify their photos on Instagram, where genetic enhancement becomes a form of self-expression, where the line between human and post-human begins to blur? These questions become even more complex when we consider the global implications of genetic engineering. Different populations have different genetic variants that affect disease risk, drug response, and other traits. Most genetic research has been conducted on people of European ancestry, meaning that gene therapies developed based on this research might not work as well for people of other backgrounds. There's a risk that genetic engineering could exacerbate health disparities rather than reducing them. Efforts are underway to make genetic research more inclusive. The All of Us research program in the United States aims to collect genetic and health data from 1 million Americans, with a focus on including populations that have been underrepresented in biomedical research. Similar initiatives are being launched in other countries, but there's still a long way to go before genetic engineering benefits all of humanity equally. The environmental applications of genetic engineering present their own set of opportunities and challenges. Scientists have developed bacteria that can break down plastic waste, potentially addressing one of our most pressing environmental problems. They've created algae that produce biofuels more efficiently than traditional crops. They've even engineered coral that can withstand higher ocean temperatures, potentially saving reef ecosystems from climate change. But releasing genetically modified organisms into the environment carries risks. Modified organisms could outcompete native species, disrupting ecosystems in unpredictable ways. Horizontal gene transfer could spread modifications to unintended organisms. The complexity of ecological systems makes it nearly impossible to predict all the consequences of releasing a genetically modified organism. The debate over genetically modified organisms, or GMOs in agriculture, has been raging for decades, and genetic engineering is adding new dimensions to this controversy. Traditional GMOs involved adding genes from one species to another, creating combinations that would never occur in nature. But new gene editing techniques like CRISPR can make changes that are indistinguishable from natural mutations. Should these be regulated differently? Are they more or less safe than traditional GMOs? In Europe, regulators have decided that gene-edited crops should be subject to the same strict regulations as traditional GMOs. In the United States, regulators have taken a more permissive approach, focusing on the traits of the modified organism rather than the method used to create it. Other countries are still developing their regulatory frameworks, creating a complex patchwork of rules that makes international trade in agricultural products increasingly complicated. Meanwhile, scientists continue to push the boundaries of what's possible with agricultural genetic engineering. They've developed wheat that can fix its own nitrogen, eliminating the need for fertilizer. They've created tomatoes with extended shelf life, reducing food waste. They've even engineered plants that glow in the dark, though that's more of a novelty than a practical application. The livestock applications are equally remarkable. Scientists have created goats that produce spider silk proteins in their milk, which can be harvested and spun into incredibly strong fibers. They've developed pigs with organs suitable for transplantation into humans, potentially addressing the critical shortage of donor organs. They've even created cattle that produce human antibodies, which can be harvested and used as medicines. But these applications raise ethical questions about animal welfare. Is it acceptable to genetically modify animals for human benefit? What if the modifications cause suffering or health problems for the animals? Where do we draw the line between acceptable use and exploitation? Different cultures and religions have different perspectives on these questions, making global consensus unlikely. The intersection of genetic engineering and artificial intelligence is opening up entirely new possibilities. AI can analyze vast amounts of genetic data to identify patterns that humans would never notice. It can predict how genetic modifications will affect an organism's traits. It can even design entirely new genes that don't exist in nature. Companies like Profluent are using AI to design novel gene editing tools that are smaller, more efficient, and more specific than naturally occurring CRISPR systems. Their AI-designed gene editors can fit into delivery vehicles that natural CRISPR systems are too large for, potentially making previously untreatable diseases accessible to gene therapy. AI is also being used to predict the effects of genetic variants on human health. By analyzing genetic data from millions of people, AI systems can identify rare variants that cause disease, predict how different people will respond to medications, and even suggest personalized treatment strategies. This combination of AI and genomics is creating a new field called precision medicine. where treatments are tailored not just to the disease, but to the individual patient's genetic makeup. The computational power required for this kind of analysis is staggering. A single human genome contains 3 billion base pairs, and analysing the interactions between different genes requires considering millions or billions of possible combinations. Only recently have computers become powerful enough to handle this level of complexity, and even now, many analyses require supercomputers or cloud computing resources. This creates its own form of inequality. Countries and institutions with access to advanced computing resources can conduct genetic research that others cannot. The digital divide becomes a genetic divide, with implications for health, agriculture, and economic development. Some worry about genetic colonialism, where wealthy countries extract genetic resources from poorer nations without sharing the benefits. Let's delve deeper into the actual experience of undergoing gene therapy, because understanding the human side of this technology is just as important as understanding the science. For patients with genetic diseases, gene therapy offers hope that was previously unimaginable. Imagine living your entire life with sickle cell disease, experiencing excruciating pain crises, organ damage, and the constant threat of stroke. Then imagine receiving a single treatment that eliminates these symptoms forever. That's the reality for patients who have received Casgevy, the CRISPR therapy for sickle cell disease. The treatment process is intense. Patients first undergo chemotherapy to destroy their existing bone marrow, making room for the edited cells. Their stem cells are harvested, edited using CRISPR to reactivate fetal hemoglobin production, and then reinfused. They spend weeks or months in the hospital while their new blood cells grow and multiply. The physical toll is significant, but for many patients it's worth it. Victoria Gray, one of the first patients to receive the treatment, went from experiencing 7 pain crises per year to none. She can now do things that were previously impossible, like picking up her children without triggering a pain crisis. She describes the treatment as giving her a new life, a chance to be the mother and person she always wanted to be. But not all gene therapy stories are success stories. There have been setbacks and tragedies along the way. In 1999, Jesse Gelsinger died during a gene therapy trial, setting the field back by years. More recently, several children receiving gene therapy for a rare immune disorder developed leukemia, though subsequent modifications to the treatment have eliminated this risk. These failures have taught valuable lessons about the importance of careful patient selection, dose optimization, and long-term monitoring. Every gene therapy trial now includes extensive safety measures, multiple review boards, and contingency plans for adverse events. The field has matured significantly, but the risks remain real. The psychological impact of gene therapy is also significant and often overlooked. Patients who have lived their entire lives with a genetic disease must adjust to being healthy. Their identity, shaped by decades of illness, must be reconstructed. Support groups have formed for gene therapy recipients, helping them navigate this unexpected challenge. For parents considering gene therapy for their children, the decision is even more complex. The treatments often work better in younger patients before permanent damage has occurred, but young children cannot consent to experimental treatments. Parents must weigh the potential benefits against the risks, making decisions that will affect their children's entire lives. The experience of pet cloning is similarly complex emotionally. People who clone their pets often report mixed feelings about the result. The cloned animal looks identical to their deceased pet, triggering powerful memories and emotions, but its personality is different, its behaviors unfamiliar. Some owners embrace the differences, seeing the clone as a continuation of their pet's legacy rather than a replacement. Others are disappointed, feeling that they've spent $50,000 on a stranger that happens to look like their beloved companion. The surrogate animals used in pet cloning also deserve consideration. These animals, often purpose, bred or obtained from shelters, carry cloned embryos to term and nurse them for the first weeks of life. They form bonds with the cloned puppies or kittens, only to have them taken away. While companies like Voyager Pets report that surrogates are well treated and adopted out after one litter, the ethical implications of using animals in this way remain contentious. The conservation applications of cloning and genetic engineering involve their own unique challenges and considerations. When scientists cloned the first black-footed ferret, They weren't just creating a single animal, they were attempting to save an entire species from extinction. The black-footed ferret population had dwindled to just 7 individuals in the 1980s, creating a genetic bottleneck that threatened the species' long-term survival. The cloned ferret named Elizabeth Ann was created from cells that had been frozen since 1988, from an animal named Willa, who died before contributing to the breeding program. Elizabeth Ann's genome contains genetic diversity that had been lost from the living population, potentially providing crucial variation for the species' survival. But integrating cloned animals into conservation breeding programs is complex. Elizabeth Ann cannot breed naturally due to reproductive abnormalities common in cloned animals, though scientists hope to use her eggs for in vitro fertilization. The success with black-footed ferrets has inspired similar efforts for other endangered species. Scientists are working to clone the northern white rhinoceros, of which only 2 females remain alive. They are attempting to create a biobank of genetic material from endangered species around the world, a frozen zoo that could preserve genetic diversity even if species go extinct in the wild. But conservation cloning faces unique challenges. Many endangered species have complex mating behaviours and social structures that are difficult to replicate in captivity. Young animals need to learn survival skills from their parents and peers. A cloned animal raised by a surrogate mother of a different species might never develop the behaviours necessary for survival in the wild. There's also the question of habitat. Saving a species' genes doesn't mean much if there's nowhere for them to live. Many conservationists argue that resources spent on cloning would be better used protecting and restoring habitats. They point out that we can't clone an ecosystem, and without healthy ecosystems, even successfully cloned animals have no future. The de-extinction projects face even greater challenges. The ecosystems that extinct species inhabited have changed dramatically. The woolly mammoth lived in a vast grassland ecosystem that no longer exists. The passenger pigeon travelled in flocks of billions, shaping forest ecosystems through their movements. Can we really bring back these species without also bringing back the worlds they lived in? Some scientists argue that de-extinction could actually help restore ecosystems. Woolly mammoths, they suggest, could help recreate the mammoth steppe by trampling snow, allowing cold air to penetrate the soil and preventing permafrost from melting. This could help combat climate change by keeping vast amounts of carbon locked in frozen soil. It's a fascinating idea, but largely theoretical. The cultural and ethical dimensions of de-extinction are equally complex. Many extinct species hold deep cultural significance for indigenous peoples. The thylacine, or Tasmanian tiger, is an important figure in Aboriginal Australian culture. Who has the right to decide whether such species should be brought back? Should indigenous communities have a say in de-extinction projects affecting species from their traditional territories? There's also the question of suffering. The first attempts at de-extinction will inevitably involve failures, animals born with deformities or health problems. Is it ethical to create suffering in the pursuit of bringing back extinct species? How many failed attempts are acceptable? These are not easy questions to answer. Let's explore the cutting edge of genetic engineering research, the technologies that are just emerging from laboratories and might transform the field in the coming years. One of the most exciting developments is the discovery of new CRISPR systems in nature. Scientists have identified dozens of different CRISPR variants, each with unique properties. Some are smaller, making them easier to deliver. Others are more specific, reducing off-target effects. Some can target RNA instead of DNA, opening up entirely new therapeutic possibilities. The discovery of CRISPR systems in organisms other than bacteria has been particularly intriguing. Scientists have found CRISPR, like systems in viruses that infect bacteria, in archaea, even in some eukaryotes. Each discovery expands our toolkit and our understanding of how nature edits genes. Researchers are also developing ways to control gene expression without editing DNA at all. Epigenetic editing involves modifying the chemical tags that tell cells which genes to turn on or off. These modifications can be temporary or permanent, heritable or not, providing exquisite control over gene expression. It's like having a dimmer switch for genes rather than just an on-off button. The field of synthetic biology is pushing even further, attempting to create entirely artificial genetic systems. Scientists have created bacteria with expanded genetic alphabets, using artificial base pairs in addition to the natural A, T, G, and C. They've developed genetic circuits that can perform computations inside living cells. They've even created minimal genomes, stripping away everything except the genes absolutely necessary for life. These synthetic biology approaches could lead to organisms designed from scratch for specific purposes: bacteria that produce valuable chemicals, algae that capture carbon dioxide more efficiently than any natural organism, even artificial cells that exist at the boundary between life and non-life. The convergence of genetic engineering with other technologies is creating possibilities that seem like magic. Scientists are developing gene therapies that can be controlled with light, activated only in specific tissues when exposed to certain wavelengths. They're creating magnetic nanoparticles that can carry genetic cargo and be guided to specific locations in the body using external magnetic fields. There's even research into using sound waves to control gene expression. Ultrasound can be focused deep inside the body without surgery, potentially allowing doctors to turn gene therapies on or off non-invasively. Imagine receiving a gene therapy that remains dormant until activated by a focused ultrasound treatment, providing precise spatial and temporal control over genetic modifications. The development of organoids, miniature organs grown from stem cells, is revolutionizing how we test genetic therapies. Scientists can now grow tiny versions of human brains, livers, intestines, and other organs in the laboratory. These organoids can be genetically modified to model diseases, allowing researchers to test treatments without risking harm to actual patients. Organoids are also being used to study human evolution. By introducing ancient genetic variants into human brain organoids, scientists can observe how our ancestors' brains might have differed from our own. They've even created hybrid organoids containing both human and Neanderthal cells, providing insights into what made us uniquely human. The combination of organoids with genetic engineering could lead to personalized medicine on an unprecedented scale. Doctors could grow organoids from a patient's own cells, test dozens of different treatments to see which works best, and then apply the optimal treatment to the actual patient. It's precision medicine taken to its logical extreme. But perhaps the most profound development is our growing understanding of how genes influence behavior and cognition. Scientists have identified genetic variants associated with intelligence, personality traits, mental illness, even political preferences. This knowledge is both powerful and dangerous, challenging our concepts of free will, responsibility, and human nature itself. The genetics of intelligence is particularly controversial. Hundreds of genes contribute to cognitive ability, each having a tiny effect, but together they account for a significant portion of the variation in intelligence between individuals. Some companies are already offering embryo selection based on polygenic scores for intelligence, though the science is far from settled and the ethical implications are staggering. What happens to our concept of merit when intelligence can be enhanced genetically? How do we maintain social cohesion when some people have been genetically optimized and others have not? These are not distant future concerns. The technology exists today, and people are already making these choices. The genetics of mental illness is equally complex and fraught. We're learning that conditions like schizophrenia, bipolar disorder, and autism involve hundreds or thousands of genetic variants, each contributing a small amount of risk. This genetic complexity makes these conditions difficult to treat with current approaches, but gene therapy offers new hope. Scientists are developing gene therapies that could adjust the levels of neurotransmitters in the brain, potentially treating depression and anxiety more effectively than current medications. They're exploring ways to modify the genes involved in neurodevelopment, possibly preventing or treating autism and intellectual disabilities. But these approaches raise difficult questions about neurodiversity and what kinds of minds we value as a society. There's something deeply unsettling about the idea of engineering the human mind. Consciousness, creativity, emotion, the very qualities that make us human, emerge from the intricate dance of genes and environment in ways we're only beginning to understand. To modify these genes is to tamper with the essence of humanity itself. Yet for people suffering from severe mental illness, these concerns might seem abstract compared to the concrete reality of their pain. If gene therapy could eliminate the hallucinations of schizophrenia, the crushing despair of depression, the chaos of mania, wouldn't it be cruel to withhold it? Where do we draw the line between treatment and enhancement, between respecting neurodiversity and alleviating suffering? The international collaboration and competition in genetic engineering adds another layer of complexity to these issues. Countries are racing to develop new gene editing technologies, to attract top scientists, to build the biotech industries of the future. This competition drives innovation but also creates pressure to move quickly, potentially at the expense of safety and ethics. China's massive investment in genetic engineering research has made it a global leader in the field. The country has more gene therapy clinical trials than any other nation and has been more willing to approve experimental treatments. This has attracted international criticism but also international collaboration, as scientists from around the world work with Chinese colleagues to advance the field. The United States remains dominant in basic research and biotechnology commercialization, home to most of the world's leading biotech companies and research institutions, but other countries are catching up. The United Kingdom has positioned itself as a leader in genomic medicine. Singapore has become a hub for synthetic biology research. Israel has developed expertise in computational genomics. This global distribution of expertise creates both opportunities and challenges. International collaboration can accelerate progress and ensure that advances benefit all of humanity. But it also makes regulation more difficult and raises concerns about biological weapons and dual-use research that could be used for harmful purposes. The potential for genetic engineering to be weaponized is a serious concern that deserves careful consideration. The same technologies that can cure diseases could potentially be used to create new pathogens. Gene drives designed to eliminate disease carrying mosquitoes could theoretically be modified to spread diseases instead. The knowledge and tools necessary for genetic engineering are becoming increasingly accessible, raising the possibility of bioterrorism. International bodies are working to prevent the misuse of genetic engineering technology. The Biological Weapons Convention prohibits the development of biological weapons, though verification and enforcement remain challenging. Scientific journals have developed policies for reviewing research that could be misused. Funding agencies require researchers to consider dual-use implications of their work. But preventing misuse while enabling beneficial research is a delicate balance. Too much restriction could slow progress on treatments for devastating diseases. Too little could enable catastrophic misuse. Finding the right balance requires ongoing dialogue between scientists, security experts, ethicists, and policymakers. The role of public opinion and social acceptance in shaping the future of genetic engineering cannot be overstated. Public attitudes toward these technologies vary widely, influenced by cultural values, religious beliefs, personal experiences, and media representation. In some countries, genetic engineering is seen as a promising solution to pressing problems. In others, it's viewed with deep suspicion. The GMO debate provides a cautionary tale about the importance of public engagement. Despite scientific consensus that GMO foods are safe, public opposition has limited their adoption in many countries. This opposition is often based on concerns about corporate control, environmental impacts, and cultural values rather than safety per se. Similar dynamics could play out with human genetic engineering. Scientists and companies are increasingly recognizing the importance of public engagement and transparency. Patient advocacy groups are being included in the design of clinical trials. Community consultations are being held before environmental releases of genetically modified organisms. Efforts are being made to explain complex science in accessible terms. But there's still a significant gap between scientific knowledge and public understanding. Surveys show that most people have heard of CRISPR but don't understand how it works. Many people conflate different types of genetic engineering, not distinguishing between somatic and germline editing or between different applications of the technology. This knowledge gap creates space for misinformation and fear-mongering. Social media amplifies extreme voices on both sides of the debate. Sensationalist media coverage focuses on designer babies and extinction, reversing while ignoring more mundane but important applications. Building public trust and understanding is crucial for the responsible development of these technologies. Education is key, but it must go beyond simply explaining the science. People need to understand the ethical, social, and economic implications of genetic engineering. They need to be equipped to participate in democratic decisions about how these technologies should be used. This requires not just science education, but also education in ethics, critical thinking, and civic engagement. The role of art and culture in shaping perceptions of genetic engineering is often underestimated. Science fiction has long explored the implications of genetic modification, from Brave New World to Gattaca to recent films and television shows. These cultural representations shape public expectations and fears, sometimes in ways that have little to do with actual science. But art can also help us grapple with the profound questions raised by genetic engineering. Novels, films, and other creative works can explore scenarios that scientists and ethicists might not consider. They can help us imagine both the benefits and risks of these technologies, to think through their implications before they become reality. There's something poetic about the fact that we're using an ancient bacterial immune system to rewrite the code of life, that we're reaching into the deep past, extracting DNA from extinct species to shape the future, that we're simultaneously playing with the building blocks of life and grappling with the most fundamental questions of existence. The pace of change in genetic engineering is breathtaking and shows no signs of slowing. Every month brings new discoveries, new applications, new ethical dilemmas. Technologies that were impossible 5 years ago are routine today. Treatments that are experimental today will be standard care in 5 years. The future is arriving faster than our ability to understand its implications. Yet, despite all this technological progress, some things remain constant. The desire to heal the sick, to protect our loved ones, to leave the world better than we found it. The fear of unintended consequences, of losing what makes us human, of creating problems we can't solve, the tension between individual choice and collective good, between innovation and precaution, between what we can do and what we should do. As we navigate this new world of genetic possibility, we need wisdom as much as knowledge, ethics as much as technology, humility as much as ambition. We need to remember that DNA is not destiny, that genes are not the whole story of who we are. We need to consider not just what genetic engineering can do for us, but what it might do to us, how it might change not just our bodies but our societies, our relationships, our understanding of ourselves. The questions we face are not easy ones. Should we eliminate genetic diseases if it means potentially eliminating certain kinds of human diversity? Should we bring back extinct species if it diverts resources from saving endangered ones? Should we enhance human capabilities if it creates new forms of inequality? Should we extend human lifespan if it exacerbates overpopulation and resource scarcity? Different people, different cultures, different nations will answer these questions differently. There is no universal consensus, no single right answer, but that doesn't mean we shouldn't grapple with these questions, that we shouldn't try to anticipate the consequences of our choices, that we shouldn't strive to use these powerful technologies wisely and well. The story of genetic engineering is still being written. We are all characters in this story, whether as patients, consumers, citizens, or simply as human beings whose future is being shaped by these technologies. The choices we make today, individually and collectively, will determine whether genetic engineering becomes humanity's greatest achievement or its greatest mistake. Perhaps the most important thing to remember is that genetic engineering is not inevitable in its current form. The technologies exist, but how we choose to use them, how we regulate them, how we distribute their benefits and risks, these are all choices. We can choose to prioritize equity over profit, safety over speed, wisdom over power. We can choose to use genetic engineering to reduce suffering and increase flourishing for all beings, not just a privileged few. As you lie there in the darkness, perhaps feeling a bit overwhelmed by the magnitude of what we've discussed, remember that you are more than your genes. You are the product of billions of years of evolution, yes, but also of your experiences, your relationships, your choices. No amount of genetic engineering can replicate the unique constellation of factors that make you who you are. And yet, we stand at a threshold. For the first time in history, we have the power to direct our own evolution, to choose not just our individual futures, but the future of our species. It's a responsibility that our ancestors could never have imagined, a power that mythology attributed to gods. How we use this power will define not just the next chapter of human history, but potentially the next chapter of life on Earth. The woolly mammoths might walk again, thundering across a restored tundra. Children might be born free from genetic diseases that have plagued humanity since before recorded history. Our beloved pets might live on in genetic duplicates, identical yet different. We might extend human lifespan, enhance human capabilities, even spread life beyond Earth using genetic engineering to adapt organisms to other worlds. Or we might create new problems we can't solve, new forms of inequality we can't overcome, new risks we can't manage. We might lose genetic diversity in our pursuit of optimization. We might create ecological disasters in our attempts to improve on nature. We might fundamentally alter what it means to be human in ways we later regret but cannot undo. The truth is, we don't know how this story ends. We're writing it as we go, making decisions whose consequences won't be fully apparent for generations. But that's always been true of human progress. Our ancestors who discovered fire, who invented agriculture, who built the first cities, they couldn't have imagined the world their innovations would create. We stand in a long line of humans who have shaped the future without fully understanding what they were doing. What makes our moment unique is the speed and scope of change. Genetic engineering is advancing exponentially, each breakthrough enabling multiple others. The time between discovery and application is shrinking. The boundaries of what's possible are expanding faster than our ability to process the implications. In this context, the question isn't whether we should pursue genetic engineering. That ship has sailed. The question is how we pursue it, with what values, what safeguards, what goals, who gets to make these decisions, who benefits from these technologies, who bears the risks. These are ultimately political and social questions as much as scientific ones. As we approach the end of our journey tonight through the landscape of genetic possibility, I want to leave you with a thought that might seem paradoxical. The more powerful our technologies become, the more important our humanity becomes. The ability to edit genes doesn't diminish the importance of compassion, wisdom, justice, and love. If anything, it makes these qualities more crucial than ever. We need scientists who are not just brilliant but also ethical. We need policymakers who understand both the science and its implications. We need citizens who are informed and engaged. We need a global conversation about what kind of future we want to create with these technologies. But most of all, we need to remember that technology is not destiny. We are not passive recipients of a technologically determined future. We are active participants in shaping that future. Every choice we make, from whether to get genetic testing to how we vote on regulations to how we talk to our children about these issues, contributes to the collective decision about how genetic engineering will transform our world. In the stillness of night, when the world softens and slows, it's easy to forget that beneath the quiet, life churns on at the molecular level. Your cells are dividing, your DNA is being transcribed and translated, errors are being made and corrected. You're a living testament to the power and fragility of genetic information, a walking library of evolutionary history and future possibility. But stillness is only an illusion, a momentary pause in the endless dance of molecules that has been going on for billions of years and will continue long after we're gone. We are temporary arrangements of atoms, brief flames in the vast darkness, yet we've discovered the code that kindles those flames. We have learned to read the book of life and are beginning to learn how to write in it. This knowledge brings both power and responsibility. We can heal diseases that have plagued humanity since time immemorial. We can bring back species from the brink of extinction, maybe even from beyond it. We can shape the future of life on Earth and perhaps beyond. But with this power comes the need for wisdom, for caution, for deep thought about what we're doing and why. Thank you for joining me on this exploration of genetic engineering and cloning, for letting me share with you the wonder and worry of this transformative moment in human history. Thank you for your curiosity, your openness to complex ideas, your willingness to grapple with difficult questions. In a world that often demands simple answers, your embrace of complexity is a gift. If this journey through the frontiers of genetic possibility has sparked your curiosity or given you new perspectives to ponder, I'd be grateful if you'd like the video and subscribe to the channel. Your support helps keep this little corner of the internet alive, this place where science and wonder meet, where complex ideas are explored with gentleness and depth. And if you're still awake, perhaps kept from sleep by thoughts of CRISPR and clones, share in the comments what aspects of genetic engineering fascinate or concern you most. Are you excited by the medical possibilities? Worried about the ethical implications? Curious about what this means for the future of humanity? I'd love to hear your thoughts. Sleep well, knowing that while you rest, scientists around the world are working to unlock the secrets of life, to develop treatments for diseases, to push the boundaries of what's possible. May your dreams be soft, your thoughts be light, and your sleep come easy, even as you carry with you the knowledge of the profound transformations happening at the smallest scales of life. And remember, this video has been for education and relaxation. Nothing we've discussed constitutes medical advice or replaces professional healthcare. If you're considering genetic testing or have concerns about genetic conditions, please consult with qualified healthcare providers. The future of genetic medicine is bright, but your health decisions should always be made with proper medical guidance. Good night, and may you rest peacefully in the knowledge that you live in an age of wonders, an age where the impossible becomes possible, where the code of life itself has become readable and writable, where the future is being invented one gene at a time. But wait, there's so much more to explore about this remarkable moment we find ourselves in. Let's delve even deeper into the intricate world of genetic manipulation, because the story we've been telling is really just the beginning of something far more complex and fascinating. Consider for a moment the actual mechanics of how cloning works at the cellular level. When scientists perform somatic cell nuclear transfer, they are essentially tricking an egg cell into believing it has been fertilized. The egg cell, stripped of its own nucleus, receives the nucleus from an adult cell, let's say a skin cell from your beloved dog. This nucleus contains the complete genetic blueprint, but it's been programmed for skin cell duties, not for creating an entire organism. What happens next is nothing short of miraculous. The egg cell's cytoplasm, that jelly-like substance surrounding the nucleus, contains factors that can reprogram the adult DNA. These factors strip away the chemical modifications that told the DNA to be a skin cell, essentially turning back the biological clock. The DNA becomes embryonic again, capable of dividing and differentiating into all the different cell types needed to build a complete organism. This process of cellular reprogramming was once thought to be impossible. Scientists believed that once a cell became specialized, once it committed to being a skin cell or a liver cell or a neuron, there was no going back. The discovery that this process could be reversed revolutionized our understanding of biology and earned Shinya Yamanaka a Nobel Prize for discovering that adult cells could be reprogrammed into induced pluripotent stem cells. The success rate of cloning remains surprisingly low, even after decades of refinement. For every successful clone born, there are dozens or hundreds of failures, embryos that don't develop properly, pregnancies that fail, offspring born with abnormalities. The process is inefficient, expensive, and ethically troubling. Yet it continues because when it works, it achieves something that seems impossible, the resurrection of a genetic identity. Think about what this means at a philosophical level. Every cell in your body, except for your red blood cells which lack nuclei, contains the complete instructions for making another you. Your skin cells, your liver cells, even the cells lining your intestines, each one holds the blueprint for your entire being. In theory, any one of these cells could be used to create your clone. You are, in a sense, a walking library of yourself, each cell a complete volume containing your entire genetic story. But here's where things get even more interesting. Not all of your cells have exactly the same DNA. As cells divide throughout your life, they accumulate mutations. Most of these are harmless, but they create what scientists call somatic mosaicism. You're not genetically uniform, but rather a patchwork of slightly different genomes. A clone made from a cell taken from your hand might be subtly different from one made from a cell taken from your foot. This genetic diversity within a single individual has profound implications for our understanding of identity and disease. Cancer, fundamentally, is a disease of somatic mutation, where one cell acquires changes that allow it to grow uncontrollably. But somatic mutations also contribute to aging, to tissue, specific diseases, even to some aspects of what makes you unique. Scientists are now developing techniques to map this somatic mosaicism, to understand how genetic diversity within an individual contributes to health and disease. They're finding that the brain is particularly prone to somatic mutations, with neurons acquiring unique genetic changes that might contribute to individuality in ways we're only beginning to understand. Let's explore the agricultural applications of cloning in more detail, because this is where the technology has been most successfully commercialized. In the livestock industry, cloning is used to replicate animals with exceptional traits. A bull that produces offspring with superior meat quality, a cow with exceptional milk production, a horse with extraordinary racing ability. These animals can be cloned to preserve their genetic value. The process is expensive, typically costing $85,000 to clone a horse, but for animals worth millions in breeding value, it can be economically justified. Some cloned animals have gone on to successful careers in competition. Several cloned horses have competed in international polo matches. Cloned bulls have sired thousands of offspring through artificial insemination. But agricultural cloning raises its own set of ethical and practical concerns. Cloned animals often suffer from health problems, including enlarged organs, breathing difficulties, and premature aging. The success rate is low, with many attempts required to produce a single healthy clone. There are also concerns about genetic diversity. If the livestock industry becomes dominated by clones of a few superior animals, it could create vulnerability to diseases that could devastate genetically uniform populations. The food safety of products from cloned animals has been extensively studied. In 2008, the FDA concluded that meat and milk from cloned animals are safe for human consumption, indistinguishable from products from naturally bred animals. Yet many consumers remain uncomfortable with the idea of eating cloned meat or drinking cloned milk, and some countries have banned or restricted these products. The European Union has been particularly cautious about animal cloning for food production. While not explicitly banned, the European Parliament has called for prohibition of cloning for agricultural purposes, citing animal welfare concerns. This creates complications for international trade, as products from cloned animals or their offspring might enter the food supply without clear labelling. Meanwhile, scientists are pushing the boundaries of what's possible with agricultural genetic engineering. They've developed salmon that grow twice as fast as normal, reaching market size in half the time. These aqua advantage salmon were the first genetically modified animals approved for human consumption, though they remain controversial and are required to be raised in land-based facilities to prevent escape into wild populations. Researchers have also created genetically modified mosquitoes designed to reduce populations of disease, carrying insects. These mosquitoes carry a gene that causes their offspring to die before reaching adulthood. When released into the wild, they mate with normal mosquitoes, gradually reducing the population. Field trials in Brazil, the Cayman Islands, and Florida have shown promising results, though concerns remain about ecological impacts. The development of gene drives takes this concept even further. A gene drive is a genetic modification that spreads through a population faster than normal inheritance would predict. Using CRISPR technology, scientists can create gene drives that could theoretically spread through an entire species within a few generations. This could be used to eliminate malaria by making all mosquitoes unable to carry the parasite or to eradicate invasive species that threaten native ecosystems. But gene drives are perhaps the most controversial application of genetic engineering. Once released, they would be nearly impossible to recall. They could spread across national borders without respect for sovereignty or consent. They could have unexpected ecological consequences, disrupting food webs in unpredictable ways. The power to reshape entire species, potentially driving them to extinction, is not one to be wielded lightly. International discussions about gene drive governance are ongoing, but have yet to produce binding agreements. Some scientists have called for a moratorium on gene drive releases until proper regulatory frameworks are in place. Others argue that the potential benefits, particularly for disease control, justify careful field trials. The debate illustrates the challenges of governing technologies that don't respect political boundaries. The synthetic biology industry is approaching genetic engineering from a different angle, treating DNA as a programming language and organisms as biological computers. Companies like Ginkgo Bioworks are engineering bacteria to produce everything from fragrances to food ingredients to industrial chemicals. They're creating biological factories that can convert sugar into products that would normally require petroleum or complex chemical synthesis. This approach to biology as engineering has attracted billions in investment. Synthetic biology companies are working on producing cultured meat without animals, spider silk without spiders, palm oil without palm trees. They're engineering algae to capture carbon dioxide and produce biofuels. They're creating bacteria that can detect and destroy pollutants in the environment. The potential of synthetic biology seems limitless, but it also raises unique concerns. Unlike traditional genetic engineering, which typically involves transferring genes between organisms, synthetic biology often involves creating entirely novel genetic constructs. These artificial genes might behave in unexpected ways. Synthetic organisms might interact with natural ecosystems in unpredictable manners. There's also the question of ownership and control. Who owns synthetic genes? Can you patent an artificial genetic sequence? What happens when synthetic organisms reproduce, carrying proprietary genetic information? These questions are being litigated in courts around the world, with implications for innovation, access, and control of genetic technologies. The DIY biology movement adds another dimension to these concerns. Community labs and biohacker spaces have made genetic engineering tools accessible to amateurs. You can buy CRISPR kits online for less than $200. YouTube tutorials teach basic genetic engineering techniques. High school students are creating genetically modified bacteria for science fairs. This democratization of biotechnology has positive aspects. It promotes scientific literacy, enables citizen science, and could lead to innovations from unexpected sources. But it also raises safety and security concerns. Amateur biologists might accidentally create harmful organisms or release genetically modified organisms into the environment. The knowledge and tools for genetic engineering are becoming so accessible that preventing misuse becomes increasingly challenging. Some biohackers are experimenting on themselves, injecting themselves with what they claim are genetic modifications for everything from muscle growth to night vision. These experiments are dangerous and unlikely to work as intended, but they represent a radical vision of human enhancement as a personal choice rather than a medical intervention. The quantified self movement has embraced genetic testing as another form of personal data. People are sequencing their genomes, tracking genetic markers over time, sharing genetic information in online communities. Some are making lifestyle choices based on genetic predictions, from diet and exercise to career and relationship decisions. This genetic self, knowledge can be empowering but also problematic. Genetic information is probabilistic, not deterministic. Having a gene variant associated with increased disease risk doesn't mean you'll definitely develop the disease. Yet people often struggle to understand these probabilities, leading to unnecessary anxiety or false reassurance. The direct-to-consumer genetic testing industry has grown rapidly, but it's also faced criticism. Some companies have made exaggerated claims about what genetic testing can reveal. Others have sold genetic data to pharmaceutical companies without clear consent from customers. The industry is largely unregulated, with little oversight of testing quality or interpretation accuracy. There's also the question of genetic privacy in the age of big data. Your genome is the ultimate personal identifier, more unique than your fingerprint, impossible to change. Once your genetic information is digital, it can be copied, shared, hacked, or subpoenaed. Laws protecting genetic privacy vary widely between countries and are often inadequate for the digital age. Law enforcement agencies are increasingly using genetic databases to solve crimes. The Golden State Killer was caught using genetic genealogy, comparing crime scene DNA to profiles in public databases. While this use has solved cold cases and brought closure to victims' families, it also raises concerns about genetic surveillance and the presumption of innocence. Insurance companies would love to have access to genetic information to assess risk and set premiums. In countries without genetic non-discrimination laws, people with genetic risk factors might find themselves uninsurable or facing prohibitive premiums. Even in countries with such laws, enforcement is difficult and loopholes exist. Employers might also be interested in genetic information. Would you hire someone with a genetic predisposition to early onset Alzheimer's for a long-term position? Would you invest in training someone likely to develop a debilitating disease? These are uncomfortable questions, but they're being asked in boardrooms around the world. The intersection of genetics and reproductive technology raises particularly complex ethical questions. Pre-implantation genetic diagnosis allows parents undergoing IVF to screen embryos for genetic conditions. This can prevent the birth of children with serious genetic diseases, but it also allows selection based on other traits. Some clinics offer sex selection, allowing parents to choose the gender of their child. This is controversial, raising concerns about gender discrimination and skewed population ratios. In some countries, sex selection is banned except for medical reasons. In others, it's freely available to anyone who can pay. The possibility of selecting for non-medical traits like eye color, height, or intelligence is technically feasible but ethically fraught. Where do we draw the line between preventing disease and enhancing capabilities? Is it acceptable to select for traits that might provide social advantages? Who decides what traits are desirable? These questions become even more complex when we consider polygenic traits, those influenced by many genes. Intelligence, personality, athletic ability. These traits involve hundreds or thousands of genetic variants, each contributing a small effect. Selecting for these traits would require making trade-offs, as genes that increase one desirable trait might increase risk for undesirable conditions. The company Genomic Prediction offers polygenic screening of embryos, providing risk scores for conditions like diabetes, heart disease, and schizophrenia, as well as predictions for traits like height and cognitive ability. Parents can use this information to select which embryo to implant. It's not quite designing a baby from scratch, but it's a significant step toward genetic selection based on complex traits. The cost of these services puts them out of reach for most people, raising concerns about genetic inequality. Will we see a future where the wealthy can afford to give their children genetic advantages while others cannot? Could this create a biological class system, where genetic enhancement becomes another form of inherited privilege? Some argue that genetic enhancement could actually reduce inequality by giving everyone the opportunity for optimal genetics, but this assumes equal access to technology that is currently expensive and likely to remain so for the foreseeable future. It also assumes we can agree on what constitutes optimal genetics, a dubious proposition given human diversity in values and preferences. The cultural dimensions of genetic engineering vary dramatically around the world. In some Asian countries, there's greater acceptance of genetic enhancement, seen as another way parents can invest in their children's future. In many Western countries, there's more emphasis on accepting genetic diversity and concerns about eugenics. Religious perspectives on genetic engineering are equally diverse. Some see it as playing God, interfering with divine will. Others view it as using God, given intelligence to reduce suffering. Some religious traditions emphasize the sanctity of natural creation. Others focus on the obligation to heal the sick and improve the human condition. Indigenous peoples often have unique perspectives on genetic engineering, particularly regarding the modification of plants and animals that hold cultural significance. The genetic modification of corn, for example, is particularly controversial in Mexico, where corn is not just a crop but a sacred element of cultural identity. The patenting of genetic sequences has been especially controversial when it involves genes from indigenous peoples or traditional crop varieties. There have been cases of biopiracy, where genetic resources from developing countries are patented by companies in developed countries without compensation or consent. This has led to calls for benefit, sharing agreements and recognition of indigenous rights over genetic resources. The Nagoya Protocol, an international agreement on access to genetic resources, attempts to ensure fair and equitable sharing of benefits from genetic resources, but implementation has been challenging, with disagreements over what constitutes a genetic resource and how benefits should be shared. Climate change adds urgency to discussions about genetic engineering. As temperatures rise and weather patterns shift, crops need to adapt quickly to new conditions. Traditional breeding might be too slow to keep pace with climate change. Genetic engineering could create drought-resistant crops, salt-tolerant plants, and varieties that can withstand extreme weather. Scientists are working on C4 rice, genetically modified to use a more efficient form of photosynthesis that could increase yields by 50%. They're developing wheat that can fix its own nitrogen, reducing the need for fertilizer. They're creating crops that can grow in marginal lands, expanding agricultural possibilities as traditional farmland becomes less productive. But critics argue that genetic engineering is a technological fix that doesn't address the root causes of climate change and agricultural problems. They point out that we already produce enough food to feed everyone. The problem is distribution and waste, not production. They worry that genetic engineering will further consolidate corporate control over agriculture, rather than empowering farmers. The role of intellectual property in genetic engineering is contentious. Patents on genes, genetic tools, and genetically modified organisms can incentivize innovation but also restrict access and increase costs. The CRISPR patent battle between the Broad Institute and the University of California, Berkeley has involved hundreds of millions of dollars and years of litigation. Some argue that genes, as products of nature, should not be patentable. Others contend that without patent protection, companies won't invest in the expensive research needed to develop genetic therapies. The balance between encouraging innovation and ensuring access remains elusive. Open source approaches to genetic engineering are emerging as an alternative to proprietary models. Some researchers are sharing genetic tools and data freely, creating commons of genetic resources. The BioBricks Foundation maintains a registry of standardized genetic parts that can be freely used for research. But open source biology also raises concerns about making dangerous knowledge too accessible. The environmental release of genetically modified organisms remains one of the most contentious aspects of genetic engineering. Once released, GMOs can't be easily recalled. They might spread beyond intended areas, potentially affecting ecosystems in unexpected ways. The precautionary principle suggests we should be extremely careful about environmental releases, but others argue that excessive caution could prevent beneficial applications. Field trials of genetically modified organisms are heavily regulated in most countries, requiring extensive safety testing and monitoring. But regulations vary widely, and there's no global consensus on what level of testing is appropriate. Some countries have embraced GMOs while others have banned them entirely. The detection and monitoring of GMOs in the environment is technically challenging. How do you track a genetically modified mosquito after it's been released? How do you detect gene flow from GMO crops to wild relatives? These practical challenges complicate regulation and enforcement. Public engagement with genetic engineering often focuses on dramatic applications like designer babies or DE extinction, but most genetic engineering is far more mundane. It's bacteria producing insulin, yeast making vaccines, plants producing more nutritious food. These applications have already transformed medicine and agriculture, largely without public notice or controversy. The normalization of genetic engineering in some domains, while it remains controversial in others, creates interesting dynamics. People who take genetically engineered insulin might oppose GMO foods. Parents who would never consider selecting their child's genes might embrace gene therapy to treat their child's genetic disease. These apparent contradictions reflect the complex ways people evaluate risks and benefits. Trust in institutions plays a crucial role in public acceptance of genetic engineering. In countries where people trust regulatory agencies and scientific institutions, there's generally more acceptance of GMOs and genetic technologies. Where trust is low, opposition tends to be higher, regardless of scientific evidence. The communication of genetic engineering science to the public remains challenging. The technology is complex, the implications are uncertain, and the stakes are high. Scientists often struggle to explain their work in accessible terms without oversimplifying. Media coverage tends towards sensationalism, focusing on extreme scenarios rather than likely outcomes. Science education about genetics and genetic engineering is improving but remains inadequate in many places. Most people learn about genetics through outdated models that emphasize genetic determinism rather than the complex interplay between genes and environment. This poor understanding makes it difficult for people to make informed decisions about genetic technologies. The role of science fiction in shaping public perceptions of genetic engineering cannot be overstated. Stories like Jurassic Park, Gattaca, and Blade Runner have created powerful narratives about the dangers of genetic manipulation. While these stories raise important ethical questions, they also create fears that might not be justified by actual science. But science fiction also inspires scientists and entrepreneurs. Many researchers in genetic engineering cite science fiction as an early influence. The vision of defeating disease, extending life, and expanding human capabilities drives much of the research in the field. The challenge is distinguishing between realistic possibilities and pure fantasy. The international governance of genetic engineering remains fragmented and inadequate. Different countries have different regulations, making it difficult to prevent regulatory arbitrage where research moves to countries with the most permissive rules. International agreements exist but often lack enforcement mechanisms. The World Health Organization has developed recommendations for human genome editing, but these are non-binding. The Convention on Biological Diversity addresses some aspects of GMOs, but doesn't cover human genetic engineering. The lack of comprehensive international governance creates risks, but also allows for diversity in approaches. Some propose a global observatory for genetic engineering, an international body that would monitor developments, assess risks, and provide guidance. Others argue for bottom-up governance, with communities and countries making their own decisions based on their values and circumstances. The tension between global coordination and local autonomy remains unresolved. The speed of technological development in genetic engineering continues to accelerate. New tools are being discovered and developed constantly. Techniques that were cutting edge 5 years ago are now routine. The pace of change makes it difficult for regulations, ethics, and public understanding to keep up. Machine learning and artificial intelligence are increasingly being applied to genetic engineering. AI can predict the effects of genetic modifications, design new genetic tools, and identify targets for gene therapy. The combination of AI and genetic engineering could accelerate progress dramatically, but also raises new concerns about control and predictability. Quantum computing might revolutionize genetic engineering by enabling simulations of biological systems that are currently impossible. Understanding how proteins fold, how genes interact, how organisms develop, these problems require computational power that classical computers can't provide. Quantum computers might unlock new possibilities in genetic engineering we can't yet imagine. The convergence of genetic engineering with other technologies creates synergies and new possibilities. 3D printing of biological tissues combined with genetic engineering could enable the creation of entirely new organs. Nanotechnology could provide new ways to deliver genetic therapies. Brain-computer interfaces might allow direct genetic control of biological processes. But technological convergence also creates new risks. The combination of genetic engineering with artificial intelligence could enable the creation of biological weapons that target specific genetic profiles. The integration of genetic information with digital surveillance could enable new forms of discrimination and control. The boundary between human and machine might blur in ways that challenge our concepts of identity and rights. The economic implications of genetic engineering extend far beyond the biotech industry. Insurance, healthcare, agriculture, manufacturing all could be transformed by genetic technologies. Countries that lead in genetic engineering could have significant economic advantages. Those that fall behind might find themselves dependent on others for critical technologies. The geopolitics of genetic engineering is becoming increasingly important. Control over genetic technologies could become a source of national power. Genetic resources could become strategic assets. The ability to engineer organisms could provide military advantages. These considerations are driving national investments in genetic research and development. But genetic engineering also offers opportunities for international cooperation. Shared challenges like pandemic preparedness, food security, and climate change could bring countries together around genetic solutions. The COVID pandemic showed how quickly genetic technologies can be deployed when there's political will and resources. The development of mRNA vaccines in less than a year was a triumph of genetic engineering. These vaccines use genetic information to instruct cells to produce viral proteins, training the immune system without using actual virus. The speed of development and deployment was unprecedented, demonstrating the power of genetic technologies to address urgent challenges. But the pandemic also revealed inequalities in access to genetic technologies. Wealthy countries secured most of the initial vaccine supply, while poorer countries waited. The knowledge and infrastructure to produce mRNA vaccines is concentrated in a few countries. These disparities raise questions about global justice and the distribution of benefits from genetic engineering. The future of genetic engineering will be shaped by choices we make today. investments in research and development, regulatory decisions, ethical guidelines, public engagement. All of these will influence how genetic technologies develop and are deployed. We're not passive observers, but active participants in shaping this future. Young people today will live in a world transformed by genetic engineering. They might have access to gene therapies that prevent aging, genetic enhancements that expand human capabilities, designed organisms that address environmental challenges. But they will also face new ethical dilemmas, social challenges, and existential questions. Education about genetic engineering is crucial for preparing future generations. Not just scientific education, but also ethical, social, and philosophical education. People need to understand not just how genetic engineering works, but also its implications for society and identity and what it means to be human. The democratization of genetic engineering could empower individuals and communities to address their own challenges. Community labs could develop organisms to clean local pollution. Patient groups could fund research into their own genetic conditions. Farmers could develop crops suited to their specific conditions. But this requires not just access to technology, but also knowledge, skills, and resources. The potential for genetic engineering to address global challenges is enormous. Malaria, which kills hundreds of thousands of people annually, could be eliminated through gene drives. Genetic diseases that cause tremendous suffering could be cured. Crops could be engineered to feed a growing population while using less water and land. But realizing this potential requires overcoming not just technical challenges, but also social, ethical, and political obstacles. Fear, mistrust, inequality, and conflicting values could prevent beneficial applications of genetic engineering. Building consensus and trust is as important as advancing the science. The story of genetic engineering is ultimately a human story. It's about our desire to understand and control nature, to reduce suffering and enhance life. It's about our fears of the unknown and unintended consequences. It's about power and justice, identity and meaning, tradition and progress. As we continue to develop and deploy genetic engineering technologies, we need to remember that we're not just editing genes, we're shaping the future of life on Earth. Every choice we make, from the research we fund to the regulations we impose to the applications we pursue, contributes to this larger story. The power to edit genes is the power to direct evolution, to choose not just our own future but the future of the biosphere. It's a responsibility that requires wisdom, humility, and deep consideration of consequences. We must proceed carefully but not be paralyzed by fear. We must be bold but not reckless. We must consider not just what we can do, but what we should do. The conversation about genetic engineering must include everyone, not just scientists and policymakers. Indigenous peoples, religious communities, young people, those living with genetic conditions, all have important perspectives that need to be heard. The future of genetic engineering should be shaped by all of humanity, not just a privileged few. We stand at a pivotal moment in history. The choices we make about genetic engineering in the coming years and decades will reverberate through generations. We have the opportunity to reduce tremendous suffering, to address existential challenges, to expand the possibilities of life. But we also risk creating new problems, exacerbating inequalities, and losing something essential about what makes us human. The path forward is not clear, and there are no easy answers, but by engaging thoughtfully with these technologies, by considering diverse perspectives, by proceeding with both courage and caution, we might navigate toward a future where genetic engineering serves humanity and the planet rather than diminishing them. The development of gene drives for malaria control represents perhaps the most ambitious application of genetic engineering to public health. Target Malaria, a research consortium, is developing mosquitoes that carry gene drives, making them unable to transmit malaria parasites or causing population collapse through biased sex ratios. The technology has shown promise in laboratory settings, with gene drives spreading through caged mosquito populations as predicted. But the step from laboratory to field release is enormous. Once released, a gene drive could spread through entire mosquito populations across Africa, potentially eliminating malaria but also causing unpredictable ecological changes. The project has invested heavily in community engagement, working with villages in Burkina Faso, Mali and Uganda to build understanding and consent for potential releases. This represents a new model of technology deployment, where affected communities are involved from the beginning, rather than having solutions imposed on them. But questions remain. How can villages in Burkina Faso meaningfully consent to a technology that could affect the entire continent? What if the gene drive spreads to countries that haven't agreed to its use? Who's liable if something goes wrong? These questions don't have easy answers, but they must be grappled with before any release. The story of Revive and Restore's work with the black-footed ferret showcases how cloning can aid conservation. By 2024, they had successfully cloned 3 black-footed ferrets from cells frozen in the 1980s. These clones, named Elizabeth Ann, Noreen and Antonia, contain genetic diversity lost from the wild population. The project required not just scientific expertise, but also regulatory navigation, as the black-footed ferret is an endangered species subject to strict protections. It also required collaboration between non-profit organisations, government agencies, zoos, and private companies. This collaborative model might be necessary for future conservation applications of genetic engineering. The cloned ferrets face their own challenges. Elizabeth Ann has reproductive abnormalities common in cloned animals. The plan is to harvest her eggs for in vitro fertilization, but success is not guaranteed. Even if breeding is successful, integrating the offspring into the wild population will require careful management to ensure the genes spread while maintaining population fitness. Let's delve into the fascinating world of xenotransplantation, the transplantation of organs from animals to humans, which has been revolutionized by genetic engineering. The shortage of human organs for transplantation is critical, with thousands dying annually while waiting for organs. Genetically modified pigs could provide an unlimited supply of organs, but the challenge has been preventing rejection and ensuring safety. Scientists have now created pigs with multiple genetic modifications to make their organs more compatible with humans. They've knocked out genes that trigger immediate rejection, added human genes that prevent blood clotting, and eliminated pig viruses that could potentially infect humans. These modifications required precise genetic engineering that would have been impossible just a few years ago. In 2022, surgeons at the University of Maryland transplanted a genetically modified pig heart into a human patient, David Bennett Sr., who was dying of heart failure and ineligible for a human heart. The heart functioned for 2 months before Bennett died, not from rejection but from a pig virus that hadn't been detected before the transplant. This case highlighted both the promise and the challenges of xenotransplantation. The genetically modified heart worked, proving the concept, but the undetected virus illustrated the complexities of ensuring safety when crossing species barriers. Subsequent transplants have incorporated lessons from this case, with more rigorous viral screening and improved genetic modifications. In 2023 and 2024, several more xenotransplants were performed, including pig kidneys transplanted into brain, dead patients for research purposes. These experiments have shown that genetically modified pig organs can function in humans for weeks or months, though long-term success remains elusive. The ethical dimensions of xenotransplantation are complex. Using animals as organ sources raises animal welfare concerns. Some religious groups object to receiving pig organs. There are questions about consent when patients are desperate and have no other options. Yet for those dying of organ failure, these concerns might seem abstract compared to the concrete possibilities of survival. The development of synthetic meat, also called cultured or lab-grown meat, represents another frontier where genetic engineering intersects with food production. Companies like Memphis Meats, now Upside Foods and Eat Just are growing real meat from animal cells without raising and slaughtering animals. The process involves taking cells from an animal, often through a harmless biopsy, and growing them in bioreactors with nutrients. Genetic engineering can optimize these cells for growth in culture, improve their nutritional profile, or enhance flavor. The result is real meat with the same proteins and fats as conventional meat, but produced without the environmental and ethical costs of animal agriculture. Singapore became the first country to approve cultured meat for sale in 2020, with Eat Just's cultured chicken nuggets. The United States followed in 2023, approving products from Upside Foods and Good Meat. The products remain expensive and limited in availability, but costs are falling as production scales up. The potential environmental benefits are substantial. Cultured meat could reduce greenhouse gas emissions, land use and water consumption compared to conventional meat production. It could also eliminate the need for antibiotics in meat production, addressing concerns about antibiotic resistance. And it could provide meat without animal suffering, addressing ethical concerns about factory farming. But cultured meat faces challenges beyond just technology and cost. Consumer acceptance remains uncertain. Some people are excited about cruelty-free meat, while others are disgusted by the idea of lab-grown food. Regulatory frameworks are still developing. The conventional meat industry is pushing back, lobbying for restrictions on labeling and marketing. There are also questions about the broader implications of cultured meat. What happens to farmers and ranchers if demand for conventional meat collapses? What about communities and cultures built around animal agriculture? How do we ensure that the benefits of this technology are distributed fairly rather than concentrating power in a few high-tech companies? The story of base editing's development illustrates how scientific breakthroughs build on each other. David Liu at the Broad Institute developed base editors by combining CRISPR's targeting ability with enzymes that can change individual DNA letters. Instead of cutting DNA and relying on error-prone repair, base editors make precise changes without double-strand breaks. The first base editors could only make certain changes, like C to T or A to G, but continued development has expanded the toolkit. There are now base editors for all possible single-letter changes, as well as prime editors that can make insertions, deletions, and replacements. These tools are already being tested in clinical trials for conditions like sickle cell disease and progeria. The development process illustrates the importance of basic research. Liu wasn't trying to cure a specific disease when he started working on base editing. He was trying to solve a fundamental problem in genome editing. But his solution has opened up possibilities for treating thousands of genetic diseases caused by single-letter mutations. The story also shows how science is increasingly international and collaborative. Liu's lab includes researchers from around the world. The technologies are being developed and applied globally. Patents and licenses are being negotiated to ensure both innovation incentives and access. The old model of science happening in isolated national silos is giving way to global collaboration, though competition remains fierce. Let's explore the world of epigenetic editing, which represents a fundamentally different approach to genetic modification. Instead of changing DNA sequences, epigenetic editing modifies the chemical tags that control gene expression. These modifications can turn genes on or off without altering the underlying genetic code. Scientists have developed CRISPR-based systems that can add or remove methyl groups from DNA, affecting gene expression. They can modify histones, the proteins around which DNA wraps, changing how accessible genes are for transcription. They can even create long-lasting changes in gene expression that persist through cell divisions. Epigenetic editing has several advantages over traditional genetic engineering. It's potentially reversible, allowing modifications to be undone if problems arise. It might be safer, as it doesn't involve breaking DNA strands. It could be more flexible, allowing fine-tuning of gene expression rather than just on-off switching. Clinical applications are being explored for various conditions. Researchers are investigating epigenetic editing for chronic pain, using it to dial down pain-signaling genes. They're exploring its use in cancer, reactivating tumor suppressor genes that have been silenced. They're even looking at applications in aging, as many age. Related changes involve epigenetic modifications. But epigenetic editing also has limitations. The modifications might not be as stable as genetic changes, potentially requiring repeated treatments. The effects might vary between cell types or individuals. The long-term consequences of epigenetic modifications are still not fully understood. The field of regenerative medicine is being transformed by the combination of genetic engineering and stem cell technology. Scientists can now take adult cells, reprogram them into induced pluripotent stem cells, genetically modify them to correct diseases, and differentiate them into needed cell types for transplantation. This approach is being used to develop treatments for conditions like macular degeneration, where genetically corrected retinal cells could restore vision. It's being explored for Parkinson's disease, creating dopamine, producing neurons from a patient's own cells. It could potentially be used to grow entire organs from a patient's cells, eliminating rejection concerns. The first clinical trials using genetically modified induced pluripotent stem cells are now underway. In Japan, researchers are transplanting genetically modified stem cell-derived retinal cells into patients with age-related macular degeneration. In the United States, trials are beginning for Parkinson's disease using genetically corrected neurons. But the technology faces challenges. Creating and validating induced pluripotent stem cells is time-consuming and expensive. Ensuring that the cells don't become cancerous after transplantation requires extensive testing. The regulatory pathway for these combination products, part cell therapy, part gene therapy, is complex and uncertain. The development of mini-organs or organoids from stem cells is opening new possibilities for personalized medicine. Scientists can create tiny versions of human organs in the laboratory, complete with multiple cell types and basic organ structure. These organoids can be genetically modified to model diseases or test treatments. Brain organoids are particularly fascinating and controversial. Scientists have created brains that develop structures resembling different brain regions, show electrical activity, and even respond to stimuli. These organoids are being used to study neurological diseases, test drugs, and understand brain development. But brain organoids raise unique ethical questions. At what point might an organoid develop consciousness or the ability to suffer? Current organoids are far too simple for such concerns, but as they become more sophisticated, these questions will become pressing. Some scientists have proposed ethical frameworks for organoid research, but consensus remains elusive. The intersection of genetic engineering and artificial intelligence is creating new possibilities we're only beginning to explore. AI can analyze vast genomic datasets to identify disease, causing mutations, predict the effects of genetic modifications, and design new genetic tools. Machine learning models can now predict protein structures from genetic sequences, a breakthrough that earned DeepMind's AlphaFold team widespread recognition. Companies like Atomwise and Recursion Pharmaceuticals are using AI to identify new drug targets based on genetic data. Others are using machine learning to optimize genetic circuits for synthetic biology applications. The combination of AI's pattern recognition capabilities with genetic engineering's precision could accelerate progress dramatically. But this convergence also raises concerns. AI systems might identify genetic modifications with unintended consequences that humans wouldn't predict. The black box nature of some AI systems makes it difficult to understand why they make certain recommendations. There are also concerns about bias in AI systems trained on genetic data from limited populations. The development of DNA data storage represents an unexpected application of genetic engineering. DNA can store information at incredible density, with all the world's data fitting in a space the size of a shipping container, if encoded in DNA. It's also stable for thousands of years under the right conditions, far longer than any current storage medium. Companies like Twist Bioscience and Microsoft are developing DNA storage systems for archival data. The process involves converting digital information to genetic code, synthesizing DNA with that sequence, and storing it. To retrieve the data, the DNA is sequenced and converted back to digital format. Current challenges include the cost and speed of DNA synthesis and sequencing, error rates in writing and reading, and the need for new computational tools to manage DNA-based data. But as these technologies improve, DNA storage could become practical for long-term data archiving. This application illustrates how genetic engineering technologies developed for one purpose can find unexpected uses. The same DNA synthesis technologies used for creating genes for therapy are being used to store cat videos and family photos. The boundaries between biology and information technology are blurring. The field of metagenomics is revealing the incredible genetic diversity of microbial life and opening new frontiers for genetic engineering. By sequencing DNA directly from environmental samples, scientists are discovering millions of new genes and thousands of new CRISPR systems, each with unique properties. These discoveries are expanding our genetic toolkit dramatically. New CRISPR variants that are smaller, more specific, or have novel functions, enzymes that can synthesize complex molecules, proteins with properties unlike anything in characterized organisms. This genetic goldmine is being mined for applications in medicine, industry, and agriculture. But metagenomics also reveals how little we know about the microbial world. We've characterized less than 1% of microbial species. The vast majority of genetic diversity remains unknown. As we engineer organisms and release them into the environment, we're operating with profound ignorance about the ecosystems we're affecting. The human microbiome, the trillions of microorganisms living in and on our bodies, is a particular focus of interest. These microbes influence our health in ways we're only beginning to understand, affecting everything from digestion to immunity to mood. Genetic engineering of the microbiome could provide new ways to treat disease and promote health. Companies are developing genetically modified bacteria as living therapeutics. These engineered microbes could produce missing enzymes in the gut, detect and destroy pathogens, or modulate the immune system. Some are already in clinical trials for conditions like phenylketonuria and inflammatory bowel disease. But engineering the microbiome is complex. The interactions between different microbial species and with the host are poorly understood. Introduced organisms might not establish themselves, or might disrupt the existing community in harmful ways. The microbiome varies significantly between individuals, making personalized approaches necessary. The development of cell-free genetic systems is enabling new approaches to biotechnology. Instead of engineering living cells, scientists are using purified genetic components in test tubes to produce proteins, chemicals, or materials. These systems can be faster, more controllable, and safer than using living organisms. Cell-free systems are being used to rapidly prototype genetic circuits, produce vaccines and therapeutics, and create biosensors for diagnostics. They can work in extreme conditions where living cells couldn't survive. They can't escape or reproduce, addressing some biosafety concerns about genetically modified organisms. The technology is still limited compared to living cells. Cell-free systems can't self, repair, or adapt. They're more expensive for large-scale production. They can't perform all the complex functions of living organisms, but for certain applications, they offer advantages that make them attractive alternatives to cellular systems. The concept of directed evolution, which won Frances Arnold a Nobel Prize, is being combined with genetic engineering to create new possibilities. Instead of rationally designing genetic modifications, directed evolution uses cycles of mutation and selection to evolve desired properties. This approach has been used to create enzymes that work in industrial conditions, antibodies that bind new targets, and organisms that produce valuable chemicals. It's particularly useful when we don't understand the relationship between genetic sequence and function well enough to design modifications rationally. New technologies are making directed evolution faster and more powerful. Continuous evolution systems can perform hundreds of generations of evolution in days. Machine learning can predict which mutations are likely to be beneficial. Microfluidics can test millions of variants in parallel. These advances are accelerating the development of new biological tools and applications. The field of genome writing, as distinct from genome editing, is emerging as the new frontier. Instead of making small changes to existing genomes, genome writing involves designing and synthesizing entire genomes from scratch. This could enable the creation of organisms with radically altered properties or even entirely new forms of life. The Genome Project Write, launched in 2016, aims to synthesize all human chromosomes within 10 years. While controversial, the project could advance our understanding of genome function and enable new therapeutic approaches. It's also driving development of DNA synthesis technologies that could have broad applications. Synthetic genomics has already achieved significant milestones. Scientists have synthesized complete bacterial genomes, yeast chromosomes, and viral genomes. Each achievement pushes the boundaries of what's possible and raises new questions about the nature of life and our ability to create it. But genome writing faces enormous technical challenges. Current DNA synthesis is expensive and error-prone. Assembling large DNA molecules is difficult. Getting synthetic genomes to function in cells requires overcoming numerous biological hurdles. The goal of writing entire mammalian genomes remains distant. though perhaps not impossible. There's something profound about the fact that we're approaching the ability to write the code of life from scratch, to create organisms that have never existed before, to explore possibilities that evolution never tried. It's a power that could enable us to address humanity's greatest challenges or create new ones we can't imagine. The regulatory landscape for genetic engineering continues to evolve as the technology advances. Different types of modifications, different organisms, and different applications fall under different regulatory frameworks, creating a complex patchwork that can be difficult to navigate. In the United States, genetic engineering is regulated by multiple agencies. The FDA oversees gene therapies and genetically modified foods. The USDA regulates genetically modified plants. The EPA regulates genetically modified organisms that produce pesticides. This division of authority can create gaps and overlaps, complicating the path to approval. The regulatory approach varies significantly between countries. The European Union has generally taken a more precautionary approach, with stricter regulations and longer approval processes. Countries like Argentina and Brazil have developed more streamlined frameworks to encourage biotechnology development. China has invested heavily in genetic engineering while maintaining tight control over certain applications. These regulatory differences create challenges for international trade and collaboration. Products approved in one country might be banned in another. Research that's permitted in one jurisdiction might be illegal elsewhere. Companies must navigate multiple regulatory systems to bring products to global markets. There's ongoing debate about how to regulate new genetic engineering techniques. Should gene editing that doesn't introduce foreign DNA be regulated differently than traditional genetic modification? How should gene drives that could spread across borders be governed? What framework is appropriate for synthetic biology applications that don't fit traditional categories? The pace of technological change is outstripping regulatory adaptation. By the time regulators understand a new technology well enough to create appropriate rules, scientists have often moved on to the next innovation. This creates uncertainty for researchers and companies while potentially allowing risky applications to proceed without adequate oversight. Public engagement in regulatory decisions about genetic engineering remains limited despite its importance. Most people have little understanding of how genetic engineering is regulated or opportunity to influence regulatory decisions. This democratic deficit undermines public trust and can lead to rejection of beneficial technologies. Some countries are experimenting with new models of governance for genetic engineering. Citizens' assemblies have been convened to deliberate on gene editing policy. Participatory technology assessment processes have engaged communities in decisions about GMO releases. These experiments in democratic governance of technology could provide models for the future. The international governance of genetic engineering remains fragmented and inadequate for technologies that don't respect borders. Gene drives, germline editing, and synthetic organisms could have global impacts, but there's no global authority to regulate them. International agreements exist, but often lack enforcement mechanisms or broad participation. Efforts to create international consensus on genetic engineering governance have had mixed success. The World Health Organization has developed recommendations for human genome editing, but these are non-binding. The Convention on Biological Diversity addresses some aspects of GMOs but doesn't cover all applications. The Cartagena Protocol on Biosafety provides a framework for transboundary movement of GMOs but has significant gaps. Some propose new international institutions or agreements to govern genetic engineering. Others argue for bottom-up governance through scientific self-regulation and national sovereignty. The tension between the need for coordination and respect for diversity in values and approaches remains unresolved. The role of intellectual property in genetic engineering continues to evolve and generate controversy. Patents on genes, genetic tools, and genetically modified organisms can incentivize innovation but also restrict access and increase costs. The balance between rewarding innovation and ensuring access to genetic technologies remains contentious.